Okay, our next speaker is Dr. Maria Hoffman. Dr. Maria Hoffman is a research genomics microbiologist at the Food and Drug Administration Center for Food Safety and Applied Nutrition, Office of Regulatory Science, Division of Microbiology. Her focus is to develop and apply microbiology and molecular genetic strategies for detecting and identifying and differentiating bacterial foodborne pathogens. So welcome, Dr. Hoffman.
Thank you. And thank you for inviting me, Chris. I'm not sure if you hear me, you just left. There. So thank you. And I'm sure you guys are wondering why I am here, because I'm doing food safety. However, we in food safety here in the last years, we actually have experienced a paradigm shift. So we have used now, since several years, whole genome sequencing successfully for surveillance and trace back for foodborne pathogens. And today I want to introduce you how we do it at the Food and Drug Administration, and maybe can inspire you how you can apply this new technology for your safety in the hospitals. I work for the Center for Food Safety and Applied Nutrition, so the FDA. So what we do, we safeguard the nation's food supply to make sure that all ingredients in the food are free of contaminants and are safe. So this is obviously not always successful, because we have in the United States about 10 million episodes of foodborne illness every year, including 60,000 hospitalizations and 1,300 deaths. And out of this, 11% are caused by salmonella. And what happens when we have an outbreak? We try to trace back the contamination very quickly. Obviously, that is not so easy, because first we have to look at the food, all the ingredients. We can go back to the facilities, back to the farms. It could have been contaminated by the animals around the farm, or maybe it got imported, the food, from other countries. So it is not. It is very difficult sometimes to do it very quickly. And to give you another perspective, we have about 200,000 registered food facilities in the United States, about 80,000 domestic and 115,000 foreign, and more than 300 ports of entry and 130,000 importers, and more than 11 million important lines each year. And also, which was make it more difficult, especially in produce, we have 2 million farms who sell their products at the market on Sundays and also at the carpets. So usually what we did was we did PFGE, pulse field electrophoresis, to trace back to the source. So as you have a restriction enzyme, it cuts the DNA. You put it out on the J electrophoresis, and you look at the different fragment sizes. However, we have seen that some strains are clonal, and sometimes the resolution was not good enough in PFGE to differentiate from an outbreak isolate to a non-outbreak isolate. So we applied whole genome sequencing. And whole genome sequencing now is not only looking at fragment sizes, which you have 10 or 11. No, we actually look at the whole genome. So we have depends how big the genome is, between 3 and 5 million data points. And what we are looking at is single nucleotide polymorphism. A single nucleotide polymorphism is a mutation between one strain to another strain, which you can see here. And the mutation differentiates one strain from another strain. The more mutation you have, the further away related you are. The less mutation you have, the more evolutionary related you are. So you can think about this as a tree, as a family tree, like from the Game of Thrones, the ancestors. So you just turn the tree around, and that's where you see sometimes our SNP trees. So it's like your ancestry when you sequence yourself on 23andMe, and you can see who are you related to, and maybe where did you come from. So the first time we used it in a retrospective study was in Salmonella Bareilles in 2012. And the Center for Disease Control and Prevention investigated a multi-state outbreak, and the federal collaborated investigative efforts from federal and state labs implicated that the yellow tuna was the source of that outbreak. And during that time, it was very difficult for us to actually trace back this outbreak to yellow tuna, because we did use PFGE, and like you can see here as an example, the non-outbreak isolates and the outbreak isolate had exactly the same PFGE pattern. And then these strains were distributed through all Southeast Asia, so we were not able to know. It made epidemiology trace back very, very difficult. So when we used whole genome sequencing, what you can see here, the green ones are very unrelated strains. So we sequenced all the Bareilles strains we had in our freezer at that time. And the one in blue had the same PFGE pattern, and then the purple ones are the clinical isolates. The orange one are the food isolates. And how you can see is that This one with the same PFGE but did not belong to the outbreak had about 117 nucleotide polymorphisms, so 117 mutations, while the one within the outbreak only had 0 to 6. So we clearly could, using whole genome sequencing, differentiate outbreak strains from non-outbreak strains. And the closest neighbor, which you can see here in blue, which goes up, where we actually saw the capability of whole genome sequencing, was an isolate which had 20 SNPs differences, and it was isolated from a shrimp 20 kilometers away from the farm, from the tuna in India. So, Uppsala. And also whole genome sequencing gives you the complete genome. So every outbreak, we complete one genome. So we know the pathogenicity islands, we know the phages, we look at the virulence genes and the antibiotic resistance genes, and we have a better understanding what we are dealing with and how to treat it. So because of the success story from Barele and how we could see that the capability of whole genome sequencing traced back even globally, we decided how cool would it be if we had a database where we collect environmental and food isolates and clinical isolates, and which we can use to trace back outbreaks even faster. So in 2013, we developed a database. So what we needed money. So we had more labs to generate the whole genome sequencing data. We needed money to actually do the network management, and we also have to store somewhere the sequence, and it has to be maintained. So luckily, NIH and NCBI, they told us, okay, we're going to do that. We're going to do this free for you. So we had more money to supply MiSeqs or sequencing instruments to the labs, and then also to generate the data and manage the data. So how does it work? So we have the labs, and they get the MiSeq, they generate the data, and immediately at the same time they generate the data, it gets uploaded to NCBI. And NCBI maintains the data and shares the data with the European molecular biologists, the NCBI in Europe, and also the Japanese one, overnight. And it gives a tree, which I'm going to show you later on overnight. And then the next day, within 24 hours, after they did their QC to make sure that it's not contaminated and everything is right, it is available for everyone. So you can download the data at the same time as we are using it to do our investigation. So at this moment, we have about 58 laboratories, which are part of our Genome Tracker database. We have state labs, obviously, but also university joins us, Penn State University and the University in Florida. We have some industry labs, like NSF is joining us, and also international labs. And you do not always have to submit through us. So we have international labs like Argentina. They send the data to us and we submit it to NCBI. But there are also international labs who are very independent, like Public Health of England is a big contributor to our database. And they do it by themselves. They just have the project linked to us with NCBI. And obviously, it is very important that the states, the federal agencies are working together. So we worked very tightly. We had an agreement in 2013 with CDC that we do this and that they are sequencing the clinical isolates and load them up in real time. And we do the same with the environmental isolates and the food isolates to get the best benefits out of that. So since then, this slide is a little bit old, it's from 2016. We have sequenced hundreds of thousands of strains. And I want to shout out to Public Health of England because they distributed over 20,000 isolates as well. And this is the pathogen detection website which you can go on at NCBI, so it's available for everyone. I have put the address down there. And there you can see how many isolates we have sequenced since then, part of our Genome Tracker database. And we have like over 80,000 of Salmonella, but also some other isolates which are from clinical settings. Because of one of our partners is actually the Brigham Hospital in Boston, Brigham Women's Hospital, it's with Lynn Bryan. And so if you, for example, click on Klebsiella here, which you see, it will load up this. And there you can see, I'm not sure if you can all see the data, but there you can see where they isolate if it has antibiotic resistance within 24 hours after you have to upload your genome. And you can also have an idea for a cluster. You can click on the isolate when you were interested in it, and you can find out more about this isolate. You can see where it's collected from. This was from the hospital in Boston. You can see when it was collected, and you can see what was the source. So we require that that we know it's a clinical, or if it's a food or environmental. Most of the times, we actually give more information. Like here, they say it's a nasal strain. And we need to know when it was isolated to get the benefit out of the database. And also, you can click on the cluster, and then it will show up your tree. So you can see which isolates are very closely related to each other. And we use this tree every single day at the core facility at the FDA to look if a clinical isolate actually match an environmental or a food isolate via sampling. So this is like an example here from an E. coli outbreak earlier this year. You can see they're very closely related, the clinical and the food isolates, only a couple snips apart. So what do we do when we know that there's a link? So I want to give you an example with Salmonella brandyrup, which was the first success story after we actually have developed this database. So in 2014, we sequenced just randomly isolates from this company which produced nut butter. And one morning, when they checked on the database if there's a match with the clinical isolates, they saw that there were 2 clinical isolates which matched exactly with this nut butter one. We immediately informed CDC, and they informed their epidemiologist. So the epidemiologist did not ask the patients now, what have you eaten in the last 2 weeks? No, they actually asked the patient, have you eaten nut butter? Which makes it much easier, since you can imagine if somebody asks you, what have you eaten last week? And you have to think about which ingredient, what kind of food you went through. And then we went back to the company to do some more sampling, and like you can see here in the tree again, they were very closely related, and the company immediately recalled their product, and not so many people got sick. So this is like, here I want to show you this. And we had other nut butter or peanut butter outbreaks the years before, and I'm not want to show that you compare the cases to the case where we used our genome tracker databases, because obviously it's a different product and you have different distribution lines. However, it does give you an idea how useful it is to really trace back an outbreak very quickly and very fast. So we had not just 700 cases in this moment where people got sick, and only 6 cases and only 1 hospitalization. Because usually what happens is if the patient gets sick, they go to the doctor, the State Department informs CDC, CDC informs us, they do the epidemiology part, and we try to trace back the food to the source, which most of the time, that's when we actually find out the source. Now, with this new technology where we actually do it at the same time, and sometimes we can even detect it earlier, like in the case of the brandyrup and the nut butter outbreak, we can interfere here and can take it out of the market. This does not only save a lot of lives, it also actually helps the industry, because early intervention means less amount of product for which can get recalled. Fewer sick patients means fewer lawsuits, and so it has a less impact overall, actually, on the damage of the brand recognition. And also it can help, and that's what I wanted to show you this slide, because I thought it would be also interesting in the clinical settings, that you know where the contamination came from. Because with sequencing, we can trace it back to the field. So you know here in this case, we had like 2 isolates, it goes through the facility, and later on we could see, okay, it's that field where the Salmonella strain came from. Or does it actually happen that it gets contaminated within the facility? So this is an approach which gives you even more than just trace back. We also can identify the species using this approach. We can know antibiotic resistance strains, so the CDC knows how to treat the patients. We know the virulence genes, and like I also mentioned, we can look for the adaptation. So we can find out maybe now why some cleaning approaches do not help. Have they actually developed quack resistance? Then at the end of the day, the industry can actually treat their facility or the strains based on scientific evidence. And it's only one approach, and it's getting cheaper and cheaper. At this moment, we are sequencing an isolate for about $40 or $50. But we do about 400, in our lab, about 400 a month. So you can see how amazing it is, and I wanted to, we have a global microbial identifier, and the director is Jürgen Schlund, and I wanted to quote him because he was saying that whole genome sequencing is the biggest thing that happened in food microbiology since Pasteur showed us how to culture pathogens. And I completely agree, and hopefully, and I know that already some hospitals, especially NIH and Lynn Bryan in Boston, and also the hospital in Germany, in Münster, they do a lot of research and sequencing isolates to find the transmission if the patients actually got sick in the hospital. Did the patient from another hospital went to this hospital and brought the strain with it? So it's all things you can find out. And if you have more information, we have a genome tracker website where you can look at it, and also a video which you can see. And obviously, this was not done only by the FDA. There's a lot of state partners, CDC, USDA involved, Public Health of England, NCBI, WHO, and all the sequencing companies who helped to make this happen. Thank you.
Okay. Thank you, Dr. Hoffman. If you could remain on stage, we'll do Q&A combined. I'd like to call up Dr. Jesse Miller, one of our phenomenal moderators for this conference.
Too kind.
And so Dr. Jesse Miller has over a decade of experience in microbiology, molecular biology, commercialization of front-end innovations. He holds a PhD in microbiology from Wayne State University School of Medicine and a BS in microbiology from the University of Michigan. In his current role at NSF International Applied Research Center, he oversees research and development activities for the organization with personal specifications in food safety and next-generation sequencing. Dr. Mo.
Thank you, John, and thank you, Chris, for the invitation to speak today. Dr. Hoffman had a great lead-in for what I'm going to talk to. I'm going to build off of her slides to kind of try to bridge that gap between food safety genomics and clinical genomics. Dr. Kelly Reynolds had a nice discussion yesterday around microbiome, right? We talked about different tools we can be using. We've had some questions about sequencing today, and other people brought it up across the board in some other talks. We do sequencing here. Dr. Hoffman does sequencing. This tool is becoming pan-available for everybody based on the driving of the cost downwards over time. So I think this is something that can be implemented in healthcare facilities, but I am not a healthcare practitioner. I'm just an idea guy. So I'm going to throw some ideas at you, get you thinking, and maybe we can have discussions at a later time about how these things can be implemented in your facility, because I don't know all the nuances of implementation in your organizations. So I'm going to tell you about next-gen sequencing. I'm going to tell you about microbiome and clinical settings. Then we're going to talk a little bit about sequencing applications. Next-gen sequencing encompasses microbiome and whole genome sequencing. So there's a 16S component as well as a whole genome sequencing component. So when you hear next-gen, it just means that's the technology we're using to get the data from these organisms. So basically, sequencing is extracting genetic material from an organism and then reading the code that you've extracted on that genetic material. I feel this is the future of health and science. Not too long ago, Craig Venter sequenced a human genome, and there was $1 million project. And like Dr. Hoffman said, now we're doing microbial genomes for $50. I'm sure you guys have seen Ancestry.com, 23andMe, these other services who will take some of your spit and tell you where you came from. What do you think they're doing? They're doing some targeted sequencing of your genome. We can sequence a whole human genome in a fraction of the time it took Dr. Venter to do it not too long ago. We can sequence microbial genomes in less than a week, sometimes in a day or two, depending on the technology you're using. We have portable technology now that fits in the palm of your hand, where I can do field sequencing out in the field. The secret to some of that is you have to do the data analysis of the sequencing, right? But you can physically do the sequencing in real time in the field, and that could be a very applicable tool for the clinical healthcare setting to get real results in real time. There's a lot of things we can use genomics for, right? You saw the Salmonella genome that Dr. Hoffman put up on the board. That genome has a lot of information in it if you know what you're looking for, right? You can look for antibiotic resistance genes, right? You can look for pathogenesis genes, pathogenicity islands, plasmids, mobile genetic elements from viruses that have gotten put inside of that genome. You can look at other propensities around that, right? What is it resistant to? Lead, arsenic, other things? What is this bug doing? What is it about once you start to look at the code, right? You can also do epidemiology. Dr. Hoffman talked about SNPs, single nucleotide polymorphisms. This is a single base pair change in a genome. This happens over time. It's part of evolution for these organisms. If you have enough of those strung together, you can start to speciate bugs and say that they're unique individuals in an environment. So what does that mean? How did they do traceback over at the FDA and Genome Tracker in that nut butter, right? They looked for some SNPs. They said, wow, this is a unique individual in this nut butter. Where else have I seen this individual? Oh, I've seen it here. Oh, gosh. Who is eating that nut butter? And then you can go out and you can stop that, right? So just think about what that could do in the healthcare setting for you if you have that level of specificity and data integrity of what you're looking at. You might have a very powerful tool right on your front doorstep that you can implement for epidemiological studies within your institution. I believe that sequencing is going to be playing a big part in personalized medicine in the future. I was just at the MBJ Summit out in California. This is a dietary supplements conference, and we're doing, you can take a drop of blood and sequence a genome nowadays, right? What happens if I sequence your genome? And I can tell you that you have genes that are generally been implicated in deficiency for something. We know that there's Alzheimer's things coming out. We have genes for a variety of other diseases. So now if you know that you have deficiencies from your genome, can I supplement that back? Can I treat you better, right? Can I give you more personalized medicine, personalized treatments to be able to help you live a more healthy and safe life so you don't get sick in the first place, right? Proactive therapy based on the knowledge of what your genome holds. It's powerful. In addition, authenticity is another thing. This is not so important for this crowd, but food safety, food fraud. There's a lot of times when things are adulterated with other materials mostly for fiscal purposes, sometimes on accident. But genomics will help you tease out what the real story is about that sample because DNA doesn't lie. So what is whole genome sequencing? This is basically the term we use for extraction of DNA from an organism and then the subsequent mapping of its genome. The genetic code gets pulled out of the organism through a variety of chemical methods, which I'm not going to belabor you with today. But there's 4 bases, right? A, G, C, T, and that makes up the whole code of the organism. Basically what happens when a sequencer runs is it reads little bits of that overall genetic code, megabases, gigabases, terabases of data, and then we assemble that using algorithms in a computer to make a linear puzzle. We put those pieces together. Some platforms, like the one Dr. Hoffman uses at the FDA, have 50,000 base pair chunks that it assembles. The one I have here in Ann Arbor is about 150 to 250 base pair chunks that I assemble. So there are different tools are out there to do the job that you need to get done, but the moral of the story is it's all the same at the end. We take pieces of the genome, read them, assemble them into a linear story, and then be able to tell you what's inside that genome. So it's basically just making a line for the data to be read later. Once you've created that code, then you can do all kinds of stuff with it, right? Tons of data analytics out there. There's tons of programs to get to answering the questions you want. Antibiotic resistance. What do the SNPs look like? How many do they have? Is it resistant to metals? What about quats? What about disinfectants? What about peracetic acid? Right? What about the things you use every day? You wonder how those bugs keep showing up. What do you think? Maybe they're getting resistant to them over time? They're not dumb. They've been here before we got here. They're going to be here after we leave, right? They want to live more than we do, maybe, right? So these guys are trying to circumvent your interventions. So by reading the genetic code, you can find out what they're doing, and maybe you have a transition in your implementation of cleaning procedures or different, materials you use to clean. So the genetic code has a lot of data that you can use. People talk about 16S metagenomics, and Dr. Reynolds talked about the microbiome. This tool takes a portion of the genome and reads it, and then it tells you what class of organisms are inside that population. So it might be Bacteroides, Firmicutes, Enterobacteriaceae, you name it, right? So it gives you a population dynamic. And then you can hit it with some sort of insult. And so it could be a different kind of cleaner, right? You could do heat treatment. You could do a bunch of water, you could do bleach, whatever. And you see how the population changes and who was able to survive through that. Dr. Ledvolk talked about biofilms the other day, right? Shishita has a nice log knockdown of the organisms inside her biofilm, but guess what? I bet you those persisters are resistant to that treatment. I bet you in their genome, you're going to find 1 or 2 guys that has some SNP change or some gene that allowed them to persist, and then they repopulate, right? Hit them again, they repopulate. Or one of their friends has something, right? How often have you seen a homogeneous biofilm? Doesn't exist. They don't exist, okay? So then those bugs start talking to each other. They start sharing genetic information and ways to survive your treatments, right? You can decipher this with genetic testing and whole genome sequencing and microbiome sequencing. You can also do shotgun metagenomics. This is another tool in the toolbox to be able to give you a little bit more speciation of the organism. For 16S metagenomics, we just look at the 16S gene, so it gives you that genus level specificity. When you do shotgun, you literally take little pieces of the whole genome of everybody inside your sample and then start to piece it together just enough to tell you who it is. Okay, it's not going to give you the single nucleotide polymorphisms and the full genome, but it's going to give you just enough to say, I know who that is. So let's say we take Dr. Chris Green. You say, Dr. Chris has a certain height. She has a certain eye color, a certain hair color. She's wearing a gray suit today. She's an organizer. You put those little pieces together. You say, oh, that's Chris Green. Nobody else in here has those things. Does that make sense? So it gives you just enough information to give you a deeper speciation on the organism, but it won't tell you differences within the species generally. So it's another powerful tool that you can use to really understand your environment. So metagenomics for your environment, okay? So what's in your environment? We talked a lot about things today with, HVAC. There's all these biofilms everywhere. There's bugs and sink traps. There's tons of reservoirs for microorganisms inside your facility. So what are we doing about it? Aeration of these organisms can be a source of infection. I pulled up this study from Cote, which we actually heard about today, right? Water comes in, splashes out, you're throwing Pseudomonas all over the place, right? So what are we going to do about that? How do you understand what's in those sink traps and what's in your environment? The clinical microbiome can help you understand that and then help you to create innovative and targeted interventions to get rid of those organisms, or at least give you more information to make data-driven decisions about it, right? You might find the certain organisms cohabitate with your bad guys, right? Maybe you have a bacillus strain or some really innocuous Hafnia strain who just likes to live in that sink trap, right? But when you knock out Hafnia, all of a sudden the biofilm that holds your Pseudomonas falls apart. That's an interesting thought, right? You don't necessarily need to kill your organism of interest. You just need to kill the other organisms that allow it to live there. So it's a dynamic environment that these bugs live in. So there's a lot of different ways you can tackle getting rid of them. Does the environment change with changes in procedures, right? What happens if you use bleach versus peracetic acid versus quats, right? What about rooms that have C. diff all the time? Does that microbiome look different? Are there different bugs that live there? Are there different resistances of the organisms that are there? What's different in that arena, right? What happens if you use something like this probiotic-based cleaning intervention where you seed a room with a bunch of beneficial organisms, right? Bugs that are good, they're not going to hurt you, and all of a sudden there's no nutrients or available spots for bad bugs to take up residence. What do you think's on our skin? A bunch of staph epidermidis, right? What do you think's in our mouth? A bunch of candida and strep mutans and other organisms. Those guys are there to protect you from the bad guys taking root. That's what happens with C. diff, right? We take antibiotics and all of a sudden C. diff's resistant to that. And now it takes over because your normal flora is not there to help you out anymore, right? So what if we start having interventions where we understand our clinical microbiome and then we can seed back beneficial organisms that protect that environment and never let the bad guys become a player? So what can you use whole genome sequencing for, right? This is where I take the whole genome of the organism. I look at every single base pair rather than just population dynamics. So epidemiology, the nut butter experiment was example was a great one from Dr. Hoffman. You have a patient come in, they're sick. Sequence that genome of that organism, right? Bank it. You understand that where it is, where it came from, right? You see another MRSA outbreak later, sequence that guy. Was it a different one? Was it the same one? You start to build a database of organisms that you've seen in your environment and inside of your facility. Then you can start to do some trend analysis and data mining to find out if that organism is a resident one from your sink trap, from your toilet seat, from the pull handle for the nurse call station, from the curtain, from the door, you name it, right? And then you can start to really be looking at stuff and say, wow, I have a bug that lives in room 22B all the time, and patients that are in 22B always seem to have this isolate when I go ahead and find out what's making them sick. Well, that's interesting. I'm going to have some sort of intervention to get that bug out of 22B, right? Or you might end up finding out, that the organism is not in 22B and it's on a healthcare worker. Or it's endemic in a little pocket right behind the desk where everybody puts their hands when they're typing on a keyboard, okay? What about antimicrobial resistance, right? This is a funny picture I'm sure you've all seen about bugs picking up something in the dark alley that helps them survive, right? A drug deal gone bad in the back there. But, what about antibiotic resistance for these organisms, VRE, CRE? We're running out of tools to be killing these bugs, right? So we're going to have to find other ways to get rid of them. But again, maybe we have more genetic information. We can make better data-driven decisions on reducing the impact of antimicrobial resistant organisms. So this is kind of the slide I really want to get to for you guys. The slides that Dr. Hoffman showed about Genome Tracker, I feel that implementation of such a platform within clinical microbiology and the clinical setting could be a very valuable tool if you poured it over with some metrics and some processes that make sense for this industry, right? It's been able to save lives, as she's shown in the FDA model. You can trace back strains very, very quickly if the data is available for everybody to see what they are. You can prevent people from dying because that strain will have antimicrobial resistance. And you don't treat it with methicillin, you treat it with something else, right? You might also end up finding out that the organisms in your facility are not really the same bugs as what you thought they were because they're a different strain or they have a different modality in which they live. So there's a lot of real good information here for epidemiological traceback to be able to understand your environment, understand what people are bringing in and not bringing in. So I think this is really important. And you do this with single nucleotide polymorphisms, right? Those are those single base pair changes that happen over time. When you have enough of those polymorphisms, you can say you have a different strain and you trace that back. This is a slide on SNPs, just kind of give you an idea what it is. I told you that, but it's a graphical representation of that single base pair change. So this is what I want to kind of talk to you about. Is there value for you in knowing who your resident strains are? Or is there risk for you in understanding who your resident strains are, right? I'm sure there's both, right? The answer is yes, Jesse. They might have antibiotic resistance you can treat better, but then you have to admit you got a bug in your environment, right? Oh, that's kind of scary, right? But what about HAI reimbursement? What if those organisms are in your facility? I have bug Bob, bug Stephanie, bug Jeremy. Those are my bugs. I own them. I got them. They're in my sink trap. I can't get rid of them. Anytime somebody gets sick, you say, do you have Bob, Stephanie, or Jeremy? Oh, you don't have Bob, Stephanie, or Jeremy? Not my bug. That's a CAI. Didn't come from me. Reimburse me, please. Not my bug. I got proof. And that's a pretty powerful tool. The genetics don't lie. The genome sequence is there, right? So does that have value for you? I don't know the answer to this question, but I want you to think about it, right? If we have a genome tracker parallel, within the clinical space. Does that have value for you? Is it risk? And how can we de-risk it if it is risky, right? There has to be ways we can get around that through data scrubbing or having some truncated data set that allows you to look at the data without having to know where it came from, perhaps. Just ideas, planting seeds. So, reimbursement is what I'm talking about. Here's another slide just to dig deeper into it. Know your risk. Can the science be communicated to prove your argument? Are there other things we can think about? And that's really what I want to leave you with today is, let's think about genomics. Let's think about it as a tool you can use as a positive tool. Let's mitigate the risk of the use of it through other means that still provide data and metrics and processes that give you value to make decisions within your organization. And maybe we can save lives and we can do it more rapidly and we can prevent infections by using genomics in your facility. Any questions? Here's my contact info. I appreciate your time.
Yeah, we'll take a couple questions if we have any. Anybody have a question for the panel?
So I'm amazed at what you just said. So chemical resistance and stuff, are we doing anything right now that we know of that is out there to combat resistance in the chemistry world?
Yeah, so we do know that some strains in fact have quark resistance genes and they carry them. However, not so much is known about their MICs. And if with a much more sanitizer, you would actually be able to kill them. So we are currently doing studies with the food industry and with sanitizers company together and with NSF actually to find out that the cleaning wasn't done right and then the sanitizer was used and that's why the strain survived, or actually did the strain really survive because of the quark resistance genes they are carrying. So we do some phenotypic and a genotypic test and the MICs. Yes, please.
So rotation disinfection, is that a value?
Yeah, that's a good question. That depends on you say rotating different products in. It would depend on the mechanism of action of that disinfectant. If it has the same mechanism of action, you could postulate that the resistance that was gained from the first product would be carried over to the second one. But that thesis is a valid one. I don't know the answer to it.
Thanks very much, panel. Very good. Quick question on resistance again, this time for emerging materials and technologies, specifically copper and UVC. I think that would be of interest to all kinds of people as the industry seems to be adopting those two things in particular. Any comment on holding?
Yeah, there's been some studies that people have been embarking on lately. So the UVC and the thymine dimers, right? The way that technology works is you're creating a bridge between two thymines, the T that I showed you before within the genome, and then the polymerase of the organism cannot make it past those two T's, so the bug therefore cannot replicate. The bug does have mechanisms by which they can try to repair that. Is it always successful? No. Sometimes you even introduce breaks into the DNA, in which case the bug's done. I think that, again, it kind of goes back to what Dr. Hoffman said with minimum inhibitory concentrations. If you don't hit them long enough, they're going to find some way to fix that and kind of bring it back. But it's a gene regulation thing. That one is pan-applicable because it is not targeting a specific enzyme or a specific process. It's targeting the DNA and damaging it. I haven't seen studies on resistances to that personally, so I can't answer your question, Brett, but I think the mechanism of action on that is different than antibiotics and some other quats and things, which it gives it a different value proposition for the user, if that makes sense.
Dr. Hoffman, Dr. Miller, may I congratulate you on your ability to simplify what is an extremely complex series of science which we're now entering, and it's just going to blow the whole world apart in terms of technology. As an old school, humble plate microbiologist, I still realize that we've still got to grow and culture these microorganisms. And if I think about one of the bugs I was talking about this morning, Mycobacterium chimera, it might take 6 weeks to grow the bacteria itself before we can do the sequencing. And also I was thinking, my colleague Samuel Collins, who did some terrific work with the PCR for Legionella, they were able to take the DNA of the PCR and then sequence type it to match it exactly to the patient strain. Some of that's beyond even my understanding, but it's terrific technology. And I just wonder where we can go with that and whether that will work with more organisms.
Yeah, I think that you do have to have a certain input of DNA to be able to run the whole genome sequencing analysis. The microbiome and metagenomics analyses need a different input of DNA. You can do a slight enrichment to try to force a reaction towards looking at an organism you would like. Oxford Nanopore, that's the handheld technology I mentioned to you before, will sequence in real time and give you real-time data, but you have to have a amount of DNA input again.
Okay, let me say something to that before, because just the new article came out. I'm sorry.
No, it's okay.
I have to interrupt you here. That where they actually amplified the DNA. So they amplified the DNA and then sequenced on the Oxford, and they got very good results out. But it is difficult. I'm trying to work with other FDA colleagues by the parasite, with Glycophoria together. Obviously they're telling me they cannot get enough DNA to sequence it because we want to get long read sequence technology, right? We can do MiSeq, which gives you 150, 200 base pairs. But to get a better genome, we want to do PacBio or Oxford, where we need a lot more DNA. So we will try the amplification and amplify the DNA before and then sequence it. How I do want to also say that still, it is very important we still have the old microbiology. No, I mean it is, and we do work together. I mean, the subtyping group in our division works together with the method development, and I work together with people who do enrichments, because we still have to know that we enrich the bacteria we want to enrich, right? Because for foodborne outbreak salmonella, I want to catch the salmonella. I'm not interested in the other ones around. So we just had a papaya outbreak, and we actually did the microbiome from the papaya to see if we can get the salmonella, and we found that the enrichment we were using did enrich the salmonella, but enriched other antibacteria as well. With listeria, it was only listeria because you have the antibiotic resistance. However, in salmonella, you enriched other bacteria as well. We were able to sequence this, and we got good results. However, there can be still stuff done. You can try to develop a better enrichment. So I'm just saying, I think it's good to work together. The same thing with epidemiology. Epidemiology is not going to die. You always need epidemiology. And I want to say before we stop the session, very quickly, sorry, we do develop an ISO standard, so we hope it comes out by the end of the year. We're meeting next week again with at the USDA together and all the international people to develop an ISO standard on whole genome sequencing.
Okay, we have time for one more quick question. Go ahead.
A question for you, Jess. I was just curious to know the genomics and PCR and all of that is a very interesting and exciting technology, but I wonder if you have a sense for the number or percentage of facilities that would be able to either perform this type of analyses or have the funding to have that type of analyses performed for them.
So there are plenty of contract organizations who will do the sequencing for you, and I can email you it's essentially a listserv website where you can punch in what you would like to have done, and there's a bunch of service providers who will do that for you. I do that here at NSF. We sequence as a service, SaaS for you software people. So we do that as well. It is a significant startup cost. Like Dr. Hoffman said, it's $50 a strain to actually sequence the organism, but your startup cost to get that machine can be upwards of $100,000. The Oxford Nanopore system is much smaller and more and cheaper to be able to start that up, but the back-end hardware you need to analyze the data can get pricey as well. And that's kind of the tricky thing when people talk about the cost of sequencing. The cost of actually running the test is cheap. The cost of analyzing the data is a different animal, right? So there's there's a lot of labs in the Genome Tracker network. I forget the exact number from your presentation, 58. So, I think that could be ported very quickly over to a clinical setting, because we definitely have the microbiology labs within these organizations to be doing microbiology. You plunk a sequencer in there. Running the sequencer is a pretty reasonable standard protocol that people can do, but then you could just send the sequencing data to somewhere else to analyze it if you have to. So I do that sometimes for very, very long read experiments. So I think you can find a way to get that done piecemeal or by contracting it out if you don't have the infrastructure to own a sequencer. I don't think that answers your question, because I don't know the full answer to your question, but I'll try it with that.
All right. Well, thank you, Dr. Hoffman and Dr. Miller.